So our next chapter we're going to discuss here is chapter 8, solution chemistry. And this chapter is really essential to understand the inner workings of the cell, because the inside and outside of the cell are just solutions. So we're going to talk about things that can be dissolved in solvents, what makes up a solution. We'll talk about colloids, things along those lines. And we're going to relate this all, of course, to biological systems. So we're going to start by discussing what is a solution. We're going to talk about solutions or mixtures. When we were breaking down matter, we had different types of mixtures. So we'll talk about our different types of solutions. We're going to talk about how solutions are formed. We're going to show some different chemical equations for solution formation. Briefly, we'll have a discussion on how we measure concentration, or how we describe concentration of different things that are dissolved in a solvent. We'll talk about dilution and how we calculate dilution factors. And this will be very important if you're, say, using a drug that you have to administer to someone that's dissolved in a stock solution. How do we cut this drug so that we can administer that in the proper dose at the proper rate for the patient? We're also going to discuss osmosis and diffusion, which is the ability for things, solutes that are dissolved in solution, to be transported across a membrane or to spread out evenly through a solution. And that of course will lead us into our transport across cell membranes, which is a very controlled mechanism for this that allows the cell to control what solutes are dissolved on the inside of the cell versus the outside of the cell. So there are a lot of different mechanisms that we'll discuss there that the body uses to keep things in a proper order. So a solution is a mixture with at least two components. It can often have many, many different components. The two major components of a solution are a solute, that's the thing that gets dissolved, and a solvent, that's the thing that does the dissolving. So one example that the book gives is a cup of coffee with sugar in it. So in this case, water is our solvent. For most biological systems, we're going to find that water is our solvent. And water dissolves the sugar. So the sugar would be the solute. That's the thing that is dissolved in the solvent. So the solvent is always going to be the thing that is present in the larger amount. So we can have many different types of solutions. Our solutions can be liquid solutions. So we can have solutions where you've got something dissolved in a liquid. An example, again, would be our coffee example. You've got the stuff from the coffee beans that dissolved in the hot water as it passed through the ground coffee in the filter. You've got the sugar in there, and you've got water. Water is the major component, so we call water the solvent. The air we breathe is another type of solution. It's a gaseous solution. In this case, we have to look at what makes up the air around us. So the air around us is roughly 79% nitrogen, N2, roughly 78% nitrogen, roughly 20.9% oxygen, 1% argon, and 0.1% other gases. So the very air that we breathe is a solution, and nitrogen is the major component of that solution. So we would call nitrogen the solvent, and oxygen would be a solute. You can have a solid solution as well. This happens with metal alloys. So we could have a mixture of two different metals. Let's say we mix like copper and aluminum together. You'd have to figure out which one is present in the highest concentration. That one would be your solvent, and the other ones would be your solute. So we can have solid, liquid, and gaseous solutions. They're all mixtures. One property of a solution is that it's transparent. You can see clear through it. So if you have something that isn't transparent, it's most likely not a solution. And we'll talk about things that might appear to be solutions but aren't in just a second. So blood is an example. Blood is a solution, but it's got other things in it that are not dissolved. So blood is a different type of mixture. A good solution is homogeneous. It's the same throughout. And we'll see that our blood is not homogeneous at all. It's very heterogeneous. You've got blood cells that are very large particles that are in your blood, and you can separate your blood by spinning it. So a solution will not separate on standing. If we dissolve sugar into water, it will not undissolve over time. It will dissolve into the water and become evenly dispersed throughout the water through a process we'll call diffusion. So if you take your sugar for a moment, that sugar will settle to the bottom of that glass. But then as it sits there over time, it's going to dissolve, assuming that we haven't put an extraordinary amount of sugar into the solution. That sugar will dissolve. It will all spread out evenly throughout the glass so that no matter where you collect a sample, the top, bottom, middle, it's going to have the same amount of sugar present because these sugar molecules are going to bump into each other and randomly disperse evenly throughout the entire mixture. So some of our most important properties of solutions, the particles are going to be evenly distributed. The components do not chemically react with one another. Not to say that they can't, but for a stable solution, there's not really a reaction going on. They're just dissolved in one another. So they're not going to chemically react. They're not going to change ionic or covalent bonds as they're sitting there until we mix multiple solutions together maybe. But there's not going to be a full-blown chemical reaction. It's just the solvent dissolves the solute. Now, in our previous chapters, we discussed these intermolecular interactions. We discussed our London dispersion forces, our hydrogen bonds, our dipole-dipole, our dipole-ion and ion-ion interactions. These are all interactions that can occur in mixtures. So for example, when you add sugar to water, there are hydroxyl groups on the sugar, as we discussed in our sugars chapter. Those hydroxyl groups can hydrogen bond with the H2O in water. So you have a hydrogen bonding interaction. So it's not a chemical reaction, but there are attractive forces that allow these molecules to dissolve in one another. If you add like motor oil to gasoline, those two share the same attractive forces, so they may form a solution. You've got London dispersion in both of those components, gasoline and motor oil. And if you mix those two components together, they will mix into a solution. And the solvent will depend on which one you added in greater volume. So if you add just a few drops of gasoline to a container of motor oil, your solvent would be the motor oil, and your gasoline would be the solute. Aqueous solutions are transparent. So all aqueous solutions, you're able to see through the solution if you had it in a glass container. That doesn't mean that it's not colored. It can be colored and still transparent. You can see what's on the other side. But all aqueous solutions are transparent. The components will not separate upon standing. If the components separate on standing, let's say we have like muddy water and the components settle out to the bottom, that's going to be something else that we're going to call a suspension. So our blood is a suspension. Once you get the components to settle out, you may have a solution remaining up top. So if we were to take the suspended material and allow it to settle, you might end up with a transparent liquid now. And that transparent liquid might be a solution. It might be something dissolved in another solvent. So that's important to recognize the difference. A suspension has larger particles suspended in it. They're not completely dissolved or broken down into individual molecules or components. In a solution, everything's broken down into individual molecules or components interacting with one another through one of these attractive forces that we discussed in the previous chapter. Another property of solutions is that you can change the concentration. So we can always change the concentration of, let's say sugar in our water. So you can continue to add more sugar to make it a more concentrated solution. We have more solute. So if you just had 1 glass of water, and let's say you had a liter bottle of water, you could add 1 gram of sugar to that. You could add 2 grams of sugar to that. That would give you different concentrations in grams per liter. So that's one of the units that we'll discuss when we discuss concentration. We're going to have a unit of mass divided by a unit of volume of the solvent. So this is going to be solvent or solute divided by the solvent is going to give us concentration. And we can measure the amounts in either mass or volume as we're describing concentrations. So just to reiterate, we've already discussed that you can have different states for our solutes and our solvents. So solutions can be a homogeneous mixture of solids, liquids, or gases. And you can have a gas dissolved in a liquid, and a liquid evaporated into a gas, so on and so forth. You can have a solid dissolved in a liquid, as is the case with our sugar dissolving into water situation, or table salt dissolving into water. We already discussed air as a perfect example. So you have other gases. So you've got nitrogen as the solvent, and your other gases are going to be the solute. If you wanted to mix a gas with a liquid, that can be done too. So our fish breathe oxygen. So there is dissolved oxygen in our water. So gases can enter the liquid phase and be a dissolved gas. So it's a gas, and that gas molecule can exit that liquid phase, especially if you heat it up and make those molecules move a little faster. That gas might want to leave. And but in order for us to survive, we have to get oxygen into our bodies. So at some point, that oxygen has to become dissolved into our bloodstream. So you can dissolve a gas. We have to get rid of our CO2, which is a byproduct of our combustion. CO2 is a gas. So we dissolve CO2 gas in our bloodstream. It hits our lungs and leaves the dissolved state back into the gaseous state, and we exhale it. So you can have transitions from a dissolved gas back into just a gas. We mentioned that you can have solid solutions. So brass is an example of a solid solution. You've got the metal zinc dissolved in another metal, copper. In this case, copper is at the higher concentration, so that's what we call the solvent. The other one, zinc, is going to be, you're going to have less zinc in brass than you have copper. So that one's going to be the solute. I mentioned the solute and solvent. They can be solid, liquid, gas. It doesn't matter. You can have one dissolved in the other. We have a special term for solutions in which water is the solvent, and we call that an aqueous solution. When we think of the term aqua, we also think of water. So any solution in which water is the solvent is an aqueous solution. Water is a really amazing compound, and we call water our universal solvent because it dissolves a large number of materials on our planet. Now, don't take that word universal and think that anything will dissolve in water, because obviously oil and water do not mix. We just call water the universal solvent because it's a very good solvent. It dissolves many more components than other solvents are able to. And that's because water can form up to 4 hydrogen bonds with neighboring water molecules. It can also interact with dipoles. It's got a dipole. It's a very strong, small polar material. Water has a really high boiling point and a really low vapor pressure, so it's not very liable to just spontaneously evaporate over time. So it creates some very stable solutions. One of the weird things about water is that its solid state is less dense than its liquid state. And that's because each water molecule can form these 4 hydrogen bonds. So when you freeze water, all of the water molecules line up and form a crystal that has a larger space between the molecules than the water does when those molecules are moving fluidly, trading partners with one another. So when water molecules line up and form this crystalline structure in which each water molecule is forming 4 hydrogen bonds, it creates this less dense material. So there's less mass per volume once it freezes. Now, if you take warm water and cool it down, you'll actually start to see that the water becomes more dense, more dense, more dense until you hit that freezing point, and then it's a reversal of that process. It expands just a little bit as it meets that freezing point, which is why ice floats in water. All other materials or most other materials on our planet, when they freeze, the molecules pack more tightly. So they increase their number of intermolecular interactions with one another, and that doesn't necessarily cause a crystalline structure to form, but they just hug each other more tightly and settle out because they're more dense. So a perfect example is when oil freezes, those molecules pack together more tightly. It's going to make a more dense solid. And if you took that frozen oil and dropped it in some unfrozen oil, some liquid oil, that would sink to the bottom. So the next thing that we're going to talk about are colloids and suspensions. So we're going to use coffee as an example. Lots of people love their coffee. And before you add your cream to your coffee, if you took your coffee and poured it into a glass mug, if it was dilute enough, you would see that that's a transparent solution. So your straight coffee that's been filtered is a solution. So you can see through that. Those materials are dissolved. Now if we take and add cream to that coffee, you'll see you won't be able to see through that solution anymore. It's become cloudy. That's because that cream, that contains particles. It contains protein particles, little chunks of protein that are too small for the naked eye to see. But those little chunks, those little tiny particles of protein, block the light as the light passes through the glass. So other materials, dissolved materials, don't block the light as the light passes through the glass. They might give it color, such as the brown or dark color of our coffee. The dissolved materials in our coffee change the color. They might change the wavelength of the light as it passes through, but they don't totally block it because they're not large enough particles to block a photon. So we have two different types of solution or mixtures that can contain particles. We can have colloids and suspensions. And the way you can tell the difference between a colloid and suspension is the size of the particles. A colloidal mixture has particles that are between 1 and 1,000 nanometers in diameter. Just to give you an idea of what a nanometer is. The nanometer is a very small measurement of length. The width of a DNA double helix, the spiral shape of DNA, is approximately 2.3 nanometers wide. So when we discuss nanometers, we're talking about things that are the size of the width of DNA, very, very small things. If you think of an E. coli cell, this is one of the simplest bacteria that we have. It's present in our gut. An E. coli cell has a width of 1,000 nanometers and a length of 2,000 nanometers approximately, or 1 micrometer and 2 micrometers. So 1,000 nanometers is equal to a micrometer. And when we discuss micrometers, we're talking about the width of bacterial cells. The width of the average human cell is somewhere between 20 to 100 micrometers in diameter. That would be 20,000 to 100,000 nanometers in diameter. So now you have an idea of what a nanometer is with respect to other parts of our body. I believe a human hair, off the top of my head, is in the several hundred micron range as far as width is concerned. So these colloidal suspensions are very small particles, and they will not separate upon standing. So if you take milk and just let it sit in your refrigerator or sit on your, if you pour it into a glass and let it sit out, it has these very small particles of protein, but they're too small to settle out by gravity, by the force of gravity. So they're suspended, very light, very suspended particles. Now, as the particles become larger, we have what's called a suspension. Suspensions contain particles that are larger than 1,000 nanometers. So particles that are 1 micrometer or larger would be suspensions. So muddy water that would separate upon standing would contain suspensions of like clay particles. So if you went down to a muddy river during a rainstorm or something along those lines, it's this brown water. If you took the sample of that and let it sit, gravity would pull down on those larger particles and they would settle out to the bottom. So you would have maybe a colloidal suspension left, maybe a solution, maybe a colloidal suspension left afterwards. So blood is a perfect example of a suspension. Blood is a suspension where we have large blood cells. These are larger than 1 micrometer in diameter, and they will settle to the bottom of a test tube upon standing. You can allow it to stand, or you can speed up this process and separate it by centrifugation. So one thing we'll do with a blood sample, if you want to look at the different components of blood, maybe run tests on the different components of blood, what you're going to have is you're going to have the blood cells are going to settle out to the bottom because they're heavier than the solution, the plasma, which is the surrounding solution that your blood cells float in. So if you have somebody that's just gone out and eaten a very fatty meal, you might get more than these 2 layers. If you just take this and separate this out, you could get on the bottom you'd have your blood, in the center you'd have your plasma, and you could have dissolved fat. Fat's an oil. It doesn't separate, it doesn't mix well with water. It's dissolved in your bloodstream as a colloidal mixture, but if you centrifuge it enough, you can get the fat to come out to the top. So you can tell if one of your patients, just got done eating like Arby's and a large milkshake or something along those lines, by how much fat would come to the top of that tube. You can tell if they've had a fatty meal before you took that sample. So we're going to talk about saturation next, because we can reach a point where there are too many solute molecules for the solvent to interact with them anymore. And we call that point the saturation point. So if we take our glass of water and add a pinch of sugar, we mentioned that that water and the sugar are going to interact with one another through these hydrogen bonds. There will come a point if we continue to add additional pinches of sugar, and we're stirring this the whole time, allowing that sugar to dissolve. If we keep adding it and keep adding it and keep adding it, we're going to find that eventually we'll reach a point where we add that next pinch, and that won't dissolve anymore. We've reached our saturation point. At this point, the water molecules are already tied up in hydrogen bonds. They're already making their maximum number of hydrogen bonds with each other and with the solute. So they can no longer break down those solid chunks of the solute. So you have a little bit of sugar left at the bottom of your glass. At this point, the solution above that solid is a saturated solution. This solution exists in an equilibrium state. So some of the sugar that's in the solution may crystallize onto the little bit of sugar that's left at the bottom. But at the same rate, you're going to have sugar dissolving back in the solution as well. So you've reached an equilibrium. The amount of solid at the bottom is not going to change over time. We're in an equilibrium state. This is just a graphical representation of what we're discussing here. So if you have a unsaturated solution, you can continue to add more and more of the solute, and the solute will continue to dissolve. Once we reach that saturation state, we will have a solid chunk of the solute at the bottom. This stuff can no longer dissolve. We'll find that some of it is dissolving and some of it is recrystallizing. This process is happening at the same rate. So because the water molecules or the solvent molecules are too tied up with other partners, they'd have to give up a partner for every new partner they take on. So that's why you can have some entering solution, some leaving solution, but they're going to happen at the same rate, those two different processes. There are several different diseases that are related to solubility. Two of those diseases are gout and kidney stones. In the case of gout, what you have is you have a buildup of uric acid in the body. Uric acid is typically cleared by the body, typically cleared by your kidneys. If this fails to happen due to your diet or other medical problems, you can have a buildup of uric acid in your body. Uric acid forms needle-like crystals. It can be a very painful thing to have happen. It happens in your joints and tendons and your cartilage. So you'll have pain and inflammation of the joints, particularly in the feet for many of these people, the feet and ankle. You'll see that, maybe in the hands. So these needle-like crystals form because your blood has become supersaturated. There's too much of the solute, in this case the solute being uric acid. And so what you're going to see, instead of that equilibrium we were just discussing, you're going to have more deposition. You're going to have the solids are going to start to build and expand because that solute is trying to leave the blood. It's too saturated. And the blood cannot interact with that anymore to dissolve any more of it. And you get a similar thing with kidney stones, which is calcium oxalate. Oxalic acid is a metabolic compound that's produced by the body. Calcium is a common ion that's dissolved in your body. And if you don't drink enough, or they're contained of more than that compound. You've got uric acid as well and calcium phosphate. So several different calcium compounds, as well as uric acid, can combine to form a kidney stone. Kidney stones are very common in people that drink very little water. If you don't hydrate well enough, if you work out and don't hydrate, or people that drink a lot of coffee. Coffee causes you to urinate, to clear the caffeine, to clear the other compounds. So you can become very dehydrated from drinking too much coffee and other caffeinated beverages. And so if you're not also drinking water along with your coffee, you're actually lowering the concentration of water in your blood. Water is the solvent in our blood. So if you have a lower concentration of water, there's not enough water to interact with the solutes, and the solutes will start to solidify again. So you're just impacting that equilibrium that we had. Now, if you add more water, more solvent, now you've diluted things, and you're less likely to have those kidney stones form in the first place. So the best way to prevent kidney stones is through increasing your intake of water, drinking a healthy quantity of water. Both gout and kidney stones can be done or can be treated through these changes in diet, and there are also some drug therapies as well that can aid in the treatment of those two diseases. So we're going to talk about what affects solubility next. How can we influence solubility, or how can we change solubility? Is there a way? We just mentioned two disease states that are related to solubility. So scientifically, can we find a way to improve the solubility of a compound? Can we get more of that compound into our solvent? Or what if we're trying to get that out of our solvent? How do we manipulate this? And we can do this by temperature, actually. It wouldn't work in the body. You can't really change a person's internal temperature. But this is very important from a biological standpoint in terms of oxygen dissolving into water. So we like to eat fish. And we're going to learn here in a second that one of the things that the Ohio EPA has done to keep our fish populations healthy is that companies that use water to cool off their processes are not allowed to discharge that water hot into our rivers and streams. And the reason is not that it's going to cook the fish. They're not just putting it out that hot, but it's because it decreases the concentration of oxygen that can dissolve in our waters. And you can suffocate your fish and other aquatic wildlife by adding warm water to a river or stream. You can change the concentration of oxygen because temperature influences solubility. Now, temperature has a different impact on the solubility of gases versus solids. So let's talk about our sugar example. If we take our little bit of sugar and drop it into our glass until we hit our saturation point, the solid chunk of sugar on the bottom is in a solid state. Those molecules are holding still. If we increase the temperature, we're speeding those molecules up. We're pushing them into a kinetic. We're increasing their kinetic energy. So if you increase the temperature, the solubility of the solids will increase. You can dissolve more sugar if you heat the solution up. The same goes with salt. If we add salt to water and you heat the solution, you can get more salt to dissolve in that solution. This is how we make candies and things along those lines. We take sugar and you're adding it to a boiling solution of water and you generate this supersaturated solution. When we cool that back off again, it's going to form sugar crystals and things along those lines. It's how we make rock candy. But while it's hot, that sugar can stay in solution. Now the opposite thing happens with gases. Gases are fast molecules. They're moving around, they're bumping around, they're in a high kinetic state, high energy kinetic state, bouncing all over the place. A gas will be more likely to enter and stay in the liquid phase if that liquid is cold. If you cool that solution off, it's going to slow that gas molecule down. If that gas molecule slows down, he's going to be less likely to fly back out of the liquid. So if you make the liquid cold, you can increase the concentration of dissolved gases in that liquid. So if you've got 20.9% oxygen in our atmosphere, and you want to increase the concentration of oxygen in dissolved water, or in oxygen that's dissolved in water, you just cool the water down. That oxygen is going to be more likely to enter and stay in that liquid water because it's getting slowed down once it enters that liquid water. It's losing its energy. It's not able to break back out. Now, if we heat that water, the opposite happens. The oxygen molecules start to move faster. And they're going to be more likely to break out of that liquid phase. They're going to be moving very rapidly, and when they hit that liquid, that transition there, the spatial transition between the liquid and the gas phase, they'll be more likely to leave because they're moving faster. So this is one of the reasons why it's very important that we monitor and not allow companies to discharge hot water back into our rivers and streams. There's a lot of companies that might do this. Power plants that generate power by boiling water. They use water to cool off things. Companies that make metals. So they've got forges and other things. They have to cool these materials down. Eventually they're using water to do this. So they have to first cool the water back down to a standardized temperature before they're allowed to discharge it back to the water body which they obtained that water from in the first place. We can also use pressure to affect solubility, and our body does this in our lungs. So when we go to exhale and inhale, we can change the pressure in our lungs, and this can change the solubility of the different gases that we're trying to take on and or release. A simple way to understand the relationship between solubility and pressure is to look at a Coke can. So we call this Henry's law of solubility and pressure. The relationship between gas solubility and pressure was summarized by this English chemist, William Henry. He found that the solubility of a gas in a liquid is directly proportional to the pressure of that gas over the liquid. So if you increase the pressure of the gas over the liquid, more of that gas is going to get forced into the liquid. That makes sense. It's very commonsensical. So our Coke cans have a high concentration of CO2. They pressurize CO2 in that Coke can. So a whole bunch of that CO2 dissolves in that sugar water, in that Coca-Cola. When you open that, you've released that pressure. So all of that dissolved CO2 is going to slowly start to bubble out of that liquid, and that's what gives it the fizziness. You're getting these new bubbles that are forming at these little points where a couple of different CO2 molecules bump into each other and they start to form a little gas bubble. And so they're leaving the liquid state when they form that little bubble. And eventually, if you leave that open to the air for long enough, your pop is now flat. It no longer has an excess of dissolved CO2 in it. So this is the system trying to re-equilibrate to the new pressure above it. The same concept is used by the body to exchange CO2 and oxygen. So the concentrations of CO2 are much higher in your bloodstream than they are in the atmosphere outside of your bloodstream. So this internal pressure of CO2 is higher in your blood. So when the blood reaches your lungs, it's going to release that CO2. Now the opposite is true for oxygen. Oxygen is in a higher concentration in the air than it is in our blood once we've used that oxygen in our metabolic processes. So when you take in a deep breath, the concentration of CO2 in your blood is higher than in the air, so the CO2 will leave. But the concentration of oxygen, the pressure of oxygen in your blood is lower than the pressure of oxygen in our atmosphere. And so that will enter your bloodstream. So this is just a graphical representation of that. When we look at our atmosphere has an atmospheric pressure of 1 atmosphere. We can split up our atmospheric pressure by splitting it up into the pressures of the individual gases that make up that total pressure. So we call that those partial pressures. So that's what the little p stands for. If you see the PCO2 and the PO2, those are the partial pressures of CO2 and oxygen in the atmosphere. And here we're showing the partial pressures of oxygen in our bloodstream. 100 millimeters of mercury versus 160 millimeters of mercury. So this is why the oxygen will enter our blood, because the partial pressure of oxygen outside in the atmosphere is higher than the partial pressure of oxygen inside. When we look at CO2, the pressure of CO2 in the outside atmosphere is 0.3 millimeters of mercury, while it's 40 in our bloodstream. So you have higher pressure of CO2 in our bloodstream than outside, so the CO2 leaves while the oxygen enters. The next thing we want to discuss when we're talking about solubility and the formation of solutions are electrolytes. Now, we're used to hearing the term electrolytes when we think of things like Gatorade or things that when we need to replenish our electrolytes, like if you get dehydrated, if you're sweating a lot, things along those lines, or if you're ill, if you have diarrhea or vomiting, you might need to replenish your electrolytes. And so let's talk about what is an electrolyte. An electrolyte is a compound that dissolves in water to form ions. So all of our ionic compounds that dissolve in water are what we would call a strong electrolyte. So things like magnesium chloride, calcium chloride, sodium chloride, a lot of things that have like a metal and then a non-metal counterion that dissolve well in water. Those are going to be our strong electrolytes. And the ones that are essential for life, you need like sodium, potassium, calcium. These are things that are involved in nerve transduction, muscle contraction, things along those lines that are essential for life. So if we deplete these in our body, we have to take them back up. And this is why we'll try to replenish electrolytes. When we put an IV in someone, there's going to be a fixed concentration of the essential electrolytes. So we're trying to match what's typically found in the blood serum to keep people healthy as we are putting fluids in them. You're not just going to put straight pure water. That would dilute the electrolyte balance in someone, and then their nerves would stop functioning, their muscles would stop functioning, their heart would stop beating. So electrolytes are essential for life, and an understanding of electrolytes is essential in the field of medicine. Our covalent compounds that do not ionize in solution, we call non-electrolytes. So are sugars. These things might still be, important for life. These are sugars. Sugars don't have a metal involved. There's no ions. There's no full charge. They interact with water in a different way. They interact with water through these attractive forces that we discussed, like that hydrogen bonding. So they don't dissociate into an anion and a cation. They interact through some other compounds. So our organic covalent compounds are what we call, that don't ionize, or we call non-electrolytes. Now you can have an organic compound that partially ionizes in water. Acids and bases will do this. When we go on to discuss acids in a future chapter, we've talked about carboxylic acid groups, and amines can act as acids and bases respectively. So these organic functional groups are what we call weak electrolytes. Some percentage of them will ionize when dissolved in water, and others won't. An example of that is shown down here on the right. You see the example of a weak electrolyte, and what we're showing here is acetic acid. Acetic acid has the formula COOH, and that represents our carboxylic acid group, shown right there. And when it has the hydrogen on it, it's neutrally charged, but when it loses that hydrogen as H+, it becomes the ion H+, and acetate ion. So acetic acid is the full CH3COOH. Acetate is the version with the negative charge that's lost its hydrogen, and you have H+ ion floating around in solution. So when you dissolve acetic acid in water, some percentage of it is going to remain as acetic acid, and some of it is going to dissociate. The percent that dissociates depends on what the pH of the solution is. So we can make the solution more acidic or less acidic and change the amount of the electrolyte or a weak electrolyte that's become an ion. So most of our weak electrolytes are going to serve as buffers in our blood. So these are going to be very important things because they can easily pick up or lose an H plus ion to change the pH in our blood. So weak electrolytes are very essential as well. All three of these are very important and serve different roles in our body. So here we're showing the chemical equation for the formation of a solution of magnesium chloride. So magnesium chloride is a strong electrolyte. And what we're doing is we're dissolving magnesium chloride in water. So as we discussed in a previous chapter, we know that this arrow means that the direction is going from left to right. And sometimes we show above or below that arrow conditions that are present. Here we're showing H2O. That means H2O is the solvent. We're not telling you how much of H2O we have. This isn't part of the chemical equation. It's not on the left side or the right side of the equation. So we're not going to balance things with respect to H2O here. H2O is just present in massive quantities compared to the magnesium and the chlorine. So what happens when you take the magnesium chloride salt and add it to water, it is going to dissociate. The magnesium ion is going to come away and become magnesium 2 plus in an aqueous state. And then 2Cl minus, this should also show that AQ for aqueous state. And what I would have shown here, I'm going to put this parentheses to the left, but it really should be to the right of the magnesium chloride, is this should have an S for solid. So we can show in parentheses to the right of the participants in the reaction, we can show what state they're in. So magnesium chloride is a solid. When you add it to water, it becomes aqueous magnesium ion plus 2 aqueous chloride ions. Just like all of our other reactions here, we've got to balance these things. So we know that magnesium and chloride will break down into their respective ions. We can tell what the charge of those respective ions are going to be based on our previous knowledge of where they're at on the periodic table of the elements. So chlorine is in group 7. It's going to have a -1 charge. Magnesium is in group 2. So it's going to have a +2 charge. So we just balance this respectively. We also know that starting out, magnesium chloride has a formula MgCl2. So that makes it really easy to balance this guy. Throwing that 2 out front of the chlorine there. And we're just going to double check our math with our charges. The charge at the beginning, you've got 2 plus for the magnesium and 2 -1s for each chlorine. So our overall charge is 0. And it's the same with our products. Overall charge is 0. So all that we're showing here is that the magnesium is no longer bound to the chloride. It's now bound to water. It's been separated from the chloride. So what's happening here is the negatively charged part of water, because remember, water is a polar molecule. It's got a partial positive here and a partial negative down here. That's going to interact with the magnesium because that's positively charged, whereas the water will flip around and the partially positively charged part with the hydrogens is going to interact with our chlorines. So water is just dissolved magnesium chloride here. This is, again, example of a strong electrolyte. All of the magnesium chloride will get converted into these ions until you've added too much magnesium chloride for the water until you've reached a saturated solution. Balancing formulas for non-electrolytes could not be easier because they don't change at all. Nothing happens to them. They just go from a solid state to an aqueous state. So, for example, we have sugar. You got your little sugar in your sugar packet, little crystals of sugar. It's a solid. You add that to water, now you have sugar water. It's in its aqueous state. So all we're changing is what's shown in the little parentheses there, your solid versus your AQ. So these are the two important groups that I discussed earlier that are going to act as our weak electrolytes. These are the most common functional groups of acids and bases in biological chemistry. You have your carboxylic acid group, and this group can act as an acid. An acid is something that donates H plus or can generate H plus in solution. We sometimes call H plus a proton. We call H plus a proton because hydrogen has a single proton and a single electron floating around that proton. If we lose that electron, all you are is a proton now. So it's H plus is just hydrogen that's lost its electron, which means it's just a proton. So carboxylic acid that loses its proton or loses its H plus is what we call a carboxylate group. On the left we have the acidic form, carboxylic acid. On the right we have the basic form, carboxylate. This reaction is reversible. It can go back and forth. So carboxylic acid can act as an acid and donate H plus, and the carboxylate can act as a base and take on H plus to go back to the carboxylic acid. So this is why this is a weak electrolyte, because this reaction is very reversible. So when you dump this stuff in water, some of it's going to lose its proton, and some of the stuff that's lost its proton might pick it back up. So that is why you won't find complete dissociation, because it's a very quick, very reversible process. The same with our amine here. Our amines, this is a very reversible process. Our amines are functional groups with nitrogen in them. Nitrogen typically has a lone pair. That lone pair can accept an H plus, which makes amines a base. Things that donate H plus are acids. Things that accept H plus are bases. And we're going to discuss that in great detail in a future chapter. Our amines are bases. They can accept an H plus. When that happens, they become a protonated amine. A protonated amine is an amine that's just picked up a proton, or H plus, which is the stuff that makes water acidic. And once that happens, that's a reversible process. So your amines may be protonated, they might not be protonated. They're going to be in some equilibrium. They're going to be bouncing back and forth. Some are going to be picking up that H plus, some are going to be losing that. So we can drive these in either direction by changing the concentration of H plus. So we can change whether that's protonated or not protonated, or whether we got carboxylic acid or carboxylate. But that's something we'll discuss in a future section when we discuss chemical equilibrium. But I want you to recognize that if you have either of these groups, the carboxylic acid or the amine, this is our weak electrolyte. So they can be in both forms, protonated or ionized or non-ionized. Just another example of the weak electrolyte, we've got acetic acid, and we already showed a picture of him. So if you start out with just acetic acid, and you add that to water, some of it is going to dissociate. As that happens, you're going to build up more and more of the dissociated ions, more and more of our products. And as the concentration of the products increases over here, that means they're going to be more likely to bump into one another and drive the reaction back in the opposite direction. As that happens, this starts to build up again, and then it's going to start to break down again until we reach a state called equilibrium, where there's no longer a change in concentration of either group. So as one is breaking down, the other is building up at an equal rate. And that rate is going to depend on the pH of the solution, or how acidic the solution is, to determine the balance of how much of each product that we have. The book throws out this definition of equivalence. When we're talking about strong electrolytes, it's important to understand what an equivalent means. And an equivalent just has to do with the charge of the stuff. So an equivalent is the amount of dissolved ion found in fluids as expressed by relative charge in a solution and the number of moles of ions present. So to really put this simply, if you have 1 mole of something with a positive 1 charge or a negative 1 charge, you have 1 equivalent. The number of equivalents goes up with the extent of the charge. So if you have a positive 2, now you have 2 equivalents. If you have 1 mole of something with a positive 2 charge, you have 2 equivalents. So if you've got 2 moles of sodium, you've got 2 equivalents. If you got 2 moles of calcium, you've got 4 equivalents. This becomes important when we're talking about ions that we may or may not know off the top of our head what their charge is. And it helps us to remember and think about how many of each ion we're going to have combined. So if we've got a solution, let's say I tell you we have a solution that's got 2 equivalents of calcium, how many equivalents of chloride am I going to need? And you would be able to say 2 equivalents. And you can figure out that means I need 2 moles of chloride. And if I asked you how much calcium did you start with in the first place in moles, you started with 2 equivalents. So you have to, because calcium has a 2 plus charge, you have to divide that by 2, so you'd have 0.5. So it's really a pretty simple, straightforward thing, the equivalence. It's just the number of moles times the degree of the charge on the ion is the equivalence. This equivalence language is often used to express concentrations of ions in blood plasma for biological systems. So you'll have a certain number of equivalents of the individual salts. And one of the reasons that we use equivalence is you don't want to change the overall concentrations of ions or ionic charge in the blood. So we speak in terms of equivalent. So let's say that you've got a constant number of positive charges in a blood sample. You could replace, if you replaced it with potassium instead of calcium, you'd only be giving, if you used equal molar concentrations, you'd only be giving them half of the charge. So the charges wouldn't add up, which wouldn't be good. And that's one of the reasons why it's important to work in these equivalents, or to understand these equivalents a little bit. So these are some of the standard concentrations of electrolytes in blood plasma with respect to milliequivalents per liter. So that's millimoles times the charge. So we don't have to always put exactly the same solution. Like we're not always trying to mimic the blood plasma for every medical application as far as fluid replacement goes. So if you got somebody hooked up to an IV, depending on their situation, you might want a different solution. And these were discovered by a lot of medical research of what is best, how patients respond to these different solutions, and a basic understanding of the physiology of the disease state or whatever is wrong with them. So one example is for replacement of fluid loss. Depending on the situation, you might want to use a sodium chloride solution or Ringer's solution. So it depends on what's going on with the patient at that point in time. You might, if it's more of a long-term thing, you might be using a Ringer's or even a maintenance solution. So if they're on a long-term IV, you have something that's probably a little more complex or complicated. You're not just trying to rehydrate the person rapidly or re-equilibrate their ions rapidly. You're trying to maintain them at their normal physiological state. So those are going to be your replacement solutions or maintenance solutions or Ringer's solutions. So concentration can be expressed in many different ways. We were just talking about concentrations in molar equivalents, which was a mole times the charge divided by a liter. So how many moles or millimoles per liter did you have? And we as chemists express things strictly as just in molarity rather than using this equivalent thing. So we'll go back. We know what the charge is on this thing, and we can do that equivalence calculation. So we just use what's called molarity. We're typically used to looking at the number of ions in solution rather than paying attention to their charge. So chemists use a term called molarity, which means moles per liter. So it's the moles of solute divided by the liters of solution, not liters of solvent. It's by liters of solution because the solute takes up space too. So that's a very important point. So if you've got 1 mole of sodium chloride divided in water and you add that to a total volume of 1 liter, you would have a 1 molar solution. Now in terms of charge equivalence, that would still be a 1 molar equivalent solution because you'd have a +1 charge on the sodium there. So it would be your equal molar equivalence. So molarity and equivalence are the same if you're dealing with things with just a singular charge. The other way that we can express concentration is as a percent of the entire solution. So we can say you've got like a 5% solution or a 10% solution. And we can do this in several different ways. It depends on what the solution's made up of. So if you've got a liquid dissolved in a liquid, and we say you've got a 5% solution, we mean a 5% solution by volume. You could also, as we discussed with our metals, when we talked about brass, you can have two metals mixed together, and so you can have percent by mass. You can also have a mass and volume percent. So one thing I want to point out before we go any further there is that when we're talking about percentages, the best units to use for percentages are the grams for mass and milliliters for volume. And the reason for that is water is going to be our most common solvent. And 1 milliliter of water weighs 1 gram. 1 milliliter of water has a mass of 1 gram. So if you are weighing a solid material in grams, it is very easy to do your math if you're working with your water in milliliters. If you're measuring your volume of your solution in milliliters, it makes the math so easy. Because 1 gram of water is the same as 1 ml, and so you don't really have to do any conversion factor problems to get between whatever unit you're using for your solute that you're measuring and the volume of the solution that you're measuring as well. So in order to calculate the percent by mass, we take the mass or volume of the solute in either grams or milliliters, whichever one you're working in, whichever is easier, and divide that by the grams or mLs of the solution, so the total solution. And we multiply that by 100 to get our percent, parts per 100. So if you had 1 gram of sodium chloride and you dissolve that into a final volume of 100 mLs, you would have a 1% solution of sodium chloride. And we'll do more examples of these calculations in our problem solving sessions. And these are more examples. So if you've got a percent by volume, you're going to have mLs over mLs. You can do mass over volume, a percent MV, which is going to be grams over mLs. So the percent thing is universally easy to do as long as you're working in grams and milliliters. Those two units work together very well. One very similar unit to percent mass per volume is a unit that's used for the measurement of hemoglobin in blood, and we use grams per deciliter. A deciliter is just 100 mLs. It's a tenth of a liter. So that's 100 mLs. If you have a 1% mass per volume solution, you have 1 per 100 mLs. So you have 1 gram per 100 mLs. And that's why this works very well. A deciliter is 100 mLs. So if you're in grams per deciliter, that's the same as saying you're in percent mass per volume. Percent means parts per 100. So if you have 1%, you have 1 gram per 100 mLs. If you have 1 gram per deciliter, you have 1 gram per 100 mLs. So it's the same exact thing. Another way that we might measure things is in parts per million and parts per trillion, and we're typically going to use this for stuff that's very dilute in solutions. Some toxins are very, very poisonous at low concentrations, so parts per million and parts per billion are going to be important numbers for them. If you look up a lot of OSHA standards for exposure limits to certain toxic gases or certain toxic materials are going to give their results in parts per million or parts per billion. So to understand a parts per billion or parts per billion, I want to go back to that example I gave where I said that 1 gram of water equals 1 ml. So 1 gram equals 1 ml. I want to point out that 1 liter is 1,000 mLs. So if I had 1 gram per liter, that would be what we call a part per thousand. Remember, 1 gram is the same as 1 ml. So if a liter is 1,000 mLs and I have 1 gram, I have 1 part per thousand. A milligram is 1,000 times smaller than a gram. So now, instead of having a part per thousand, I have a part per million. If we multiply 1,000 by 1,000, we get a million. So I've added the milli on there. So it made it 1,000 times smaller. So 1 milligram is a part per million. If you have 1 microgram per liter, now you have a part per trillion. So we might say ppm, parts per million. That's going to refer to 1 milligram per liter. And that means for every molecule of water, or for every million molecules of water, you've got 1 molecule of whatever is dissolved. So you have 1 milligram of that material per liter of water. And parts per billion is 1 microgram per liter. So you've got, for every billion parts of water, you've got 1 part of whatever is dissolved. A really important concept when we're talking about concentration is going to be dilution. Especially in the medical field, you might have a concentrated drug that you might have to dilute before you administer to someone. We're going to have to know how to do the math to do this. Or you might have a solution that you need to use to prep something, and it might come as a stock. Now, the reason a lot of these things come as a stock is sometimes they're more stable at higher concentration. So they're shipped concentrated to remain stable. Sometimes it's just cheaper to ship them that way. Everybody's got tap water, and if you're dissolving it in water, then it's really easy to add water. It would be expensive to ship that water across country from wherever that drug was synthesized or whatever it is that you're injecting into the person. So things often will come as a stock solution in order to save shipping costs. They're concentrated. Perfect example is orange juice. Orange juice comes concentrated. Everybody's got tap water in their kitchen, so it's not very difficult to take this concentrated orange juice, dump it into a thing, and add cans until you get the concentration that you want. How dilute do you want your orange juice? So that's just the same thing that we're going to learn how to do here with different drugs. So when we're doing our dilutions, one thing that's very important to remember is that the amount of dissolved material is not changing. The concentration might change, but the amount of material stays the same. When we add that whole can of orange juice and then add water to it, the contents of that can of orange juice aren't going anywhere, right? They're not disappearing, but they're getting diluted. They're being spread out through a larger volume of material. So the concentration in that total volume is going to decrease. So this is our formula for calculating dilution factors. If we need to make something from a stock concentrated solution, how do we make it? How are we going to do this? And the formula just has 4 simple parts. It has the initial concentration, which is C initial, the initial volume, which is V initial. These are the concentration and volume of the stock solution that you need. So then say we're going to administer a certain concentration and volume to a patient, that would be the final concentration and the final volume. So typically we're going to be given the concentration of our initial solution. We're going to be told the final concentration that we want and how much of it we want. And we're going to have to solve for this initial volume. So what we'll do then is we'll figure out what the initial volume we need is, and we'll add water to get to the final volume. So that's all we're really doing here. How much water do we need to add to how much of our stock solution? That's what we're looking for. So we'll do some examples of that. C initial, V initial equals C final, V final problem or equation in our problem solving sessions. And we're going to move on here and just discuss dilution factors as concentrated stock solution. Sometimes they make it really easy for you. If the solution is always used at the same concentration, they might just tell you it's a 4x stock solution, which would mean that you just have to dilute it by 4 times of whatever volume you're using. Or they can make a 5x or a 10x or a 20, any x that they want. So if there's always a constant concentration of the stuff used, and a perfect example would be media to grow cells in for cellular biology stuff, you're always going to be keeping the same concentration of electrolytes on the outside of the cell. Some of your, if for some reason a solution that you were going to inject into somebody to keep their blood stable, you want a stable concentration of ions, that might come as a stock solution. Most often those materials, to avoid contamination, are going to come already fully loaded, or they're prepared in a lab at the hospital, and they're going to be shipped up to the nurses or whoever's going to be administering them fully loaded, and you're not going to be doing the dilution on something that you're going to be injecting to somebody. But if you were, these ones that come showing you this 4x or 2x or whatever, makes it very easy. If it's a 4x stock solution, you just have to dilute it by 4 times, which would mean you want the final volume to be 4 times higher. So if you had that can of orange juice and it's a 4x solution, you would put the can of orange juice into the container. That's 1. Then you would use 3 additional cans of water. You've just multiplied the volume by 4 times by doing that. So let's say that was a 100 ml can of orange juice. If it's a 4x solution, then your final volume should be 400 mLs. So you'll add your can of orange juice in there, then you'll fill up that water can until you've reached that 400 mLs point. One of the final things we're going to discuss in this chapter is osmosis. Osmosis is the ability for ions to travel across what we call a semi-permeable membrane. Our cell membrane is a semi-permeable membrane. This is a membrane which allows some molecules to pass through, but not others. There are other examples of semi-permeable membranes. We can make membranes out of things like cellulose, some of our plastics, and make the pores so small that certain things can cross but other things cannot. So you could make it so that ions can cross but water molecules cannot. Or water molecules can cross but big protein molecules and things along those lines cannot. So you've got something that's a semi-permeable. Some things can permeate it, some things cannot. Our cell is the cell wall. It's a perfect example of a semi-permeable membrane. Under normal physiological conditions, the concentrations of ions on both sides of the cell membrane are the same, but this can become perturbed, either by the body or by ingesting the wrong thing or by injecting the wrong thing into someone. We could perturb this, what we call an isotonic solution. Isotonic meaning they're equal concentrations or equal equivalents of ions on either side. It doesn't mean that you have the same number of sodium ions on both sides of the membrane. It means you have the same number of total ions. So it's very common for the human cell to have, say, elevated levels of sodium on the outside, potassium on the inside of the cell. So it does sort things out, but you're going to have an equal concentration of ions. When you count all the ions up, it should be constant on both sides. If not, we're going to have problems, and we'll see that in a second. So you can actually kill yourself by drinking too much water. When you drink too much water, you're taking in large quantities of the solvent without the solutes. Regular tap water does not have as many dissolved ions as our bloodstream does in it. So what you do when you drink too much water is you actually create a hypotonic solution outside of your cells. Hypo means you have less than, and tonic is our salts or our ions. So we've got or other dissolved components. So if you are hypotonic, you have a more dilute solution than what is typically found in the cell. So water can travel across the cell membrane And it's going to do that to try to restore equilibrium. Remember, things were at an equilibrium earlier. Your body's got an equal concentration of salts and other dissolved materials on the inside and outside of the cell. So if you add a whole bunch of water and dilute that, the water is going to try to move towards the region with the higher concentration of dissolved materials. So if we add a hypotonic solution to the cells, the inside of the cell is going to be more concentrated than the outside of the cell with respect to dissolved materials. So water is going to rush into the cells to try to dilute that, to try to reach an equilibrium again. And if this happens, the cells can swell up and burst. So we call this process cell lysis. And I've got a figure for that in just a moment. So this process literally generates pressure on the cells. We discussed pressure earlier in this chapter. And this literally adds pressure to our cells. The pressure is what we call the osmotic pressure. So the greater the difference in concentration between the two solutions on opposite sides of the membrane, the higher the osmotic pressure is going to be. So you can actually fight osmotic pressure by applying physical pressure in the opposite direction, and that will stop the osmosis. We actually utilize this to purify water. We do the reverse of this. We'll take a semi-permeable membrane and we'll reverse the process. We can take a semi-permeable membrane that can only allow water to pass through and not allow ions to pass through. And if you apply enough force, you can get the water across. So this is one way that we can purify drinking water from salt water. Speaking of salt water, if we add too much salt, this is why we can't drink salt water. We talked about drinking tap water earlier, which would have a lower concentration of salts in it than our bloodstream does. But you can have a hypertonic solution as well, something with too much dissolved salt. In the case of too much dissolved salt, you're going to have excess salt on the outside of the cell. Therefore, the water on the inside of the cell is going to rush out of the cell to try to dilute that excess salt on the outside. So this is going to result in the cells becoming dehydrated or shrinking. So this is a great little picture that shows you what's happening in these different situations here. So if we take a cell and we add it to an isotonic solution, the isotonic solution means that there's an equal concentration of salts on the inside as the outside, and nothing happens to the cell. Perfectly normal, happy little cell. If we take a hypotonic solution, so you've got fewer particles on the outside than you do on the inside. You got more and more particles on the inside than you do on the outside here. So what's going to happen is the water is going to try to neutralize that situation. So the water can either move into or out of the cell. Remember, the salts can't move. I've got the salts shown as the little red and yellow dots. The salts are not free to cross that membrane, but water can. If water left the cell in this case, it would just make the situation even worse. The cell would shrink and the things would get more concentrated. So water instead is going to try to dilute. It's going to go into the cell. So that's why if you have a hypotonic solution, you got this hemolysis. We have the opposite thing going on here. With our hypertonic solution, we have many salt, way more salt outside of the cell than we do inside of our cell in this case. And so what's going to happen is, in order to try to reach equilibrium again, the water is going to leave the cell. And it's going to try to dilute that salt on the outside. And when that happens, we get the cell shrink and you get what we call crenation. So you can have hemolysis versus crenation, both of which can be very lethal to the cell and eventually lethal to the person. So now that we understand that, we understand why it's very important that we only use the right materials when you're trying to rehydrate someone. If you're giving someone something intravenously, it's very important that we maintain that isotonic solution outside of the cell. So all of our intravenous solutions
that we inject into people's bloodstreams are going to be isotonic. So one of the more common ones are that you've got that 0.9% sodium chloride, 0.5% glucose solution. It's called D5W. And this is an example what we call a physiological solution. When you say physiological solution, that's synonymous with it's isotonic to human blood, so we can administer this and keep the patient healthy. So now we've discussed osmosis. We're going to discuss diffusion and dialysis, which are slightly related to osmosis. We were discussing equilibriums with osmosis, and the system's going to attempt to reach an equilibrium in which things are equally concentrated on all sides. So all these water molecules have a fair equal number of partners to interact with. That's really one of the driving forces behind this osmosis and the diffusion. It's the water molecules fighting over binding partners. So they prefer to be bound to these dissolved things. This is a strong interaction that they have. So that's what drives this. That's why there's actual physical pressure. There's a physical driving force. It's the strength of these interactions that we discussed, those hydrogen bonding interactions or the ion dipole interactions that are driving these things. And the same thing happens if you just take like a drop of, let's say, food color, for example. If I put a drop in a beaker, it's not going to be very long before all the water molecules have forced each other to equally share that dye throughout the entire beaker. They're going to try to interact with that, and just through random bumping into each other, it's all going to get equally spread throughout the solution. So we call this process diffusion. So you can start with a very concentrated little drop. If you drop it into a bigger volume, it's going to diffuse throughout the entire solution. And that's going to be useful for what we call dialysis here. So a very good medical example would be for kidney dialysis. Our kidneys are little dialyzing machines. They're really cool. They use diffusion to move waste molecules from one direction to another across these membranes. So they have these membranes and they keep the concentrations different one side than the other. And so what happens is your small waste molecules will move out of the blood through diffusion across membranes in the kidneys, and that can be removed as waste. So our small molecules will diffuse out of the blood and move into the urine, which is in a lower concentration than the blood. And this process is called dialysis. If that process fails, we can undergo an artificial dialysis that we call hemodialysis. It's used to cleanse the blood. So in this process, we take our blood, remove it from the patient, pass it through one side of a semi-permeable membrane in contact, and on the opposite side of that is an isotonic dialyzing solution. So things like urea and other small waste molecules are going to diffuse out of that while they're passing through that isotonic dialyzing solution, and then the dialyzed purified blood is going to return back to the patient. Again, this is done in patients that have failing kidneys, where their kidneys are not able to undergo this dialysis properly on their own. And this is just an example of what that system might look like. You'd have your dialysis chamber. So you've got the patient hooked up to an IV, you've got the blood coming out, blood going back in. You've got some pumps that are keeping this going. And you've got a dialyzing coil, which has a semi-permeable membrane. So the small molecule waste products and things along those lines can leave the blood, enter this dialyzing solution, and then that is properly disposed of. We mentioned that water can travel freely across the cell membrane, and that's not because water can go right through that lipid bilayer. It's because there are channels in that cell membrane that water can pass through. The lipid bilayer is pretty difficult to get through. It's got that charged outside, that very polar, those polar head groups, and a very hydrophobic inside. That makes it a pretty impermeable membrane. There are very few molecules. There are molecules out there that can do it, but there aren't that many that can interact with both polar things and non-polar things. There are molecules out there that can do it. We talked about like our cholesterols and things along those lines that have polar and non-polar groups. There are solvents like dimethyl sulfoxide is an example of things that can easily permeate the cell membrane. And so those things can be used to like transport toxins on purpose, to kill people. But typically, our cell wall acts as a very strong barrier, and it doesn't allow things inside or out of the cell unless it's done so on purpose via what we call facilitated or active transport, which these are going to be done by integral membrane proteins. So these are going to be proteins that are stuck inside of the cell wall. So we really have three different ways that things can get inside or out of our cell wall. We can have passive diffusion. And like I said, that can happen if the molecule is able to interact with both the hydrophobic and hydrophilic parts of the cell wall. So some molecules have the ability to passively diffuse across the cell wall. Other molecules are going to have perhaps a channel. So a facilitated transport means there's a facilitator. There is someone that is selecting, okay, yes, you are the right size ion. You must be potassium. I am going to let you through now. So there's a protein that has that job. It can be done by active transport, which requires energy. So let's say the cell is a muscle cell and it contracts. When a muscle cell contracts, calcium is released into the cell, and that triggers the contraction event. Now, when it wants to relax again, it has to pump that calcium back into vesicles or back outside of the cell, one of the two. So that's going to be done by an active transport mechanism, which is going to lower the concentration. So this is working against the osmotic gradient. So active transport requires energy. The facilitated transport is going to be working with the osmotic gradient. So one example might be there's excess sodium on the outside of the cell versus the inside of the cell, and the cell wants to let a little bit of sodium in. That's not a problem. There's less sodium on the inside, so the sodium is going to go across its osmotic gradient to try to reach an equilibrium. So it's going to easily pass through if there's a channel there that it can fit through. So some examples of things that can go through that passive diffusion are going to be small molecules like oxygen, O2, nitrogen, N2, CO2. These things can directly diffuse through the cell membrane. So they're going to move so that they reach equal concentrations on either side of the cell membrane in a process that we call passive diffusion. There are some larger molecules that can also do this. Like we said, our cholesterols, our steroids, things that have both a very big hydrophobic region and then a polar head group may be able to do this as well. As we said, that facilitated transport is another way that we can get things across a membrane. So if something could not go across that membrane normally, there may be a integral membrane protein that can enable that small molecule to pass through that membrane. So this facilitated transport does not require energy, and the cell can open and close that channel. So if the cell wants to stop letting that small molecule in, it could potentially close that channel to stop that from happening. And then let's say it wants to let some more back in, it can open that channel back up and allow for this facilitated transport to continue to occur. So let's say we want to increase the concentration of something and go against the osmotic gradient. Let's say that we've got, like, we want to make a really high concentration of acid, like on the inside of the stomach per se. We need an active transport mechanism. So we would need something that we can fight osmosis with, something we'd have to use energy to get this to work. So active transport does that by burning ATP, adenosine triphosphate. This is the energy molecule of the body, or the energy currency of the body. An example of this is the transport pump, the potassium hydrogen ATPase pump, K plus H plus ATPase pump. This controls the concentrations of potassium and hydrogen ions in the stomach, which makes your stomach acidic so that we can digest our food. Now, people that have acid reflux disease, where the acid doesn't stay in their stomach, it comes up into their esophagus. We might want to decrease the concentration of acid in their stomach. This is not good for their digestion at all. It's sometimes they have to take supplements to make sure that they're getting the right nutrients and things along those lines, because we are purposefully perturbing a natural process, supposed to prevent them from getting their esophagus from eroding and getting esophageal cancer. So we could take something that blocks these active transport proteins. So the drugs Tagamet, Zantac, Pepcid, there are several other stronger ones, are examples of drugs that would inhibit this active transport and therefore slow down the rate at which the cells can create acid inside of the stomach. And that's just one example of active transport. There are many other examples of active transport in biology. Well, that's it for chapter 8. So just to review a little bit, we discussed what a solution was. We said that a solution was a mixture, and we'd like you to be able to distinguish between the solute and the solvent. We'd like you to be able to identify what is a solution versus a colloid and a suspension. Those are three different things, and you should know how to differentiate between the three of them. We're going to talk about the formation of solutions. We want to talk about a saturated solution, so you should be able to discuss that. What is a saturated solution? What effect does temperature have on the solubility of solutes? What effect does pressure have on the solubility of gases in a liquid? Pressure particularly has an effect on gases. Temperature can have a different effect on solids and gaseous solutes too, so keep that in mind too when you're reviewing that material. Additionally, we discussed chemical equations for solution formation, so I just want you to recognize that transition from solid to aqueous state. That's really an important takeaway there. And being able to get from equivalents to moles and vice versa. Again, very simple. You're just multiplying or dividing by the charge on the ion. You should be able to calculate concentration of materials and molarity, which is you're just going to take moles divided by liters. And again, we discussed percent and parts per million, parts per billion. These are things that we covered earlier in chapter 1. We also went over dilution, so you should be able to use that dilution formula. And again, we are going to do some examples of that in our problem solving session in class, so that C initial, V initial equals C final, V final equation, very important there. We should also know what osmosis is, be able to predict what's going to happen to a cell if you put that cell in an isotonic, hypotonic, or hypertonic solution, and then know the three different types of transport across cell membranes.